Before I do the research thing, I'm going to give my 5-minute, 3-minute advertising spiel. Tyrone. All right. It's good to see old friends from the days that I was here. So I am now a pseudo-administrator. If you can call administration running a field station, that's the kind of administration you might want to do someday. It's a great gig. I spend the summers, I get to migrate and live at a field station with my family and a bunch of bio geeks, and it's like a summer camp. So a lot of you have been to field stations. If you ever need one in the East Coast, the Mid-Atlantic states, this is a good place to come, Mountain Lake. We're at the top of a mountain. This is our western view across West Virginia. It's not on site, it's just down the road. Beautiful climate. It's not that nasty, sticky East Coast summer that you think it might be. It's truly beautiful, and it is in the center of plethodontid diversity in North America. We have a couple different things that we run. I'm just going to blitz over a few things. We support independent research. We do courses, so if you're a grad student looking for specialized training in some area, we may be able to cover you. And we run an RU program. The research that we do is not, it's not station-based, it's not programmatic, it's not the kind of thing that long-term ecological research sites. We cater to individual researchers who want to do research there, and we try to do whatever we can to facilitate that research. So if you've got questions about what we do, what we have, what the facilities are, that's my job, is to answer those questions. The courses are standard field station fare. They range from Chris Schneider, some of you know him. Manhandling one of our timber rattlers, showing the herpetology class and everyone else who wanted to get scared rattlesnake. We have graduate, undergraduate classes. Last year we had a wildlife disease class that was taken by a lot of grad students from other places. Systematics of fungi. So there's, you just have to watch the site to see what we're doing this year. The one thing I draw your attention to if you're a grad student, this isn't, you won't find it advertised anywhere, but talk to us if you want to take a class and you don't care about credit. You want the training, but you don't want the letter grade on your transcript, because we can make costs much more reasonable in those cases. And we get spectacular instructors, like Chris. His training's a little suspect. And then RU sites. If you guys have undergraduates, if there are undergraduates here that are looking for a field experience, you know about RU sites in various places. We have 10 to 11 a year. We have about 250 applicants. It's incredibly competitive. They get paid lots of money to come and do science and live in a cool place and pal up with mentors. So if you're looking for a place in the East, holler. It's a great place to play. So now, science. I'm going to talk about a long-term collaborative project that kind of started before I was here as a postdoc. I've drug it on longer than I expected with my most frequent collaborator, who's also my dad. As you can guess, there's some mixed benefits and costs to that relationship. We look at the coevolutionary interaction between a predator and prey. I'll talk about the general aspects of it and then where we've headed most recently. So we'll start with some common ground. What do we mean by coevolution? When we're talking about it, essentially the natural history, what we call a phenotypic interface. What traits are important in driving coevolution, and why do they matter to this interaction? We'll talk about what we spent a long time, I guess the last maybe 5-8 years on, and that's geographic structure and geographic mosaics, and what role do subdivided populations have in driving the dynamics. And then what we've focused on most recently is the molecular genetics of resistance. So trying to understand at a reductionist level, really, what are the mechanisms that generate resistance, and what can we tell about evolutionary process when we get down to that level. So a little disclaimer, this is not a place I ever saw myself going as a scientist, and I have to admit I'm pretty jazzed by it now. So most of you guys are on this page already, but if you talk about any coevolutionary interaction, let's just take antagonism here between hosts and parasites, reciprocity is what matters. So you've got selection generated by one species that drives adaptation in that second species. The fact that derived character state or change in phenotype happens in that other species increases selection back on the original species, and they in turn evolve. Right? So that doesn't necessarily present a particular dynamic, it just presents reciprocity. And that, I think, is one of the things that's missing in a lot of coevolutionary studies, probably including our own. Right? Why do people get excited about it? There are a couple of specific things you should keep in mind. When we talk about any kind of antagonistic system in coevolution, we really have to have a couple of things. We have to have reciprocal interactions that stem from that ecological interaction. I think this is the weakest part of predator-prey systems that have been characterized this way, and I'll say more about that in a minute. And you have to have the genetic potential to respond to that selection. So in order to talk about coevolution, we have to have the ecological factors, the genetic complementarity. And that means specifically that we've got genetic basis and variation to the traits that matter. Reason people get excited, several reasons. Really rapid evolution results from this. So you imagine this as not a classic runaway system, but essentially the same sort of dynamic where selection feeds on itself and makes itself stronger. That generates really rapid phenotypic change. As a result, probably, we get specialized interactions in a lot of cases, and people have modeled most recently the spin-off of speciation processes after this specialization. So this may be a major source of biodiversity. It's been claimed to be one of the reasons that we get so much genetic diversity in the tropics. And then there are all kinds of cool dynamics. So if you're into Red Queen cycles or arms races of escalation and counter-escalation, that's where these things come from. So why did I badmouth predator-prey interactions? If you go through all the papers that say we're studying predator-prey arms races, about 98% of them study some elaborate anti-predator mechanism. Very cool. I like anti-predator mechanisms, but they aren't really looking at a two-way street. They're really showing dramatic responses of prey to predators. Conceptually, people have criticized this on an asymmetry level, so there's selection asymmetries that are expected between predators and prey. Dawkins and Krebs called this the life dinner principle. So in an interaction between two species, this Leptophis has much less to lose in the interaction than the Norops does, obviously. Norops loses the interaction, zero fitness. Norops loses a meal. The other aspect that weakens selection on predators is something that we call dodging the bullet. And this is essentially a variance statement. The predictability of that consequence to this horned lizard was complete. If it loses life, again, a zero effect. There's really not much variance in the consequence for a loss. But for the shrike that killed this lizard, if it had missed the lizard, the consequence of missing that meal depends on its own condition, what the available prey base is, whether it's feeding young in a nest. The variation in what that realized consequence is large. High variance generates weak selection. Weak consequences generate weak selection, and both of those are expected to be more prevalent on the predator side than the prey side. That's why people think there's an asymmetry. So we spent a fair amount of time looking at this saying, well, why do we see predators evolve resistance in the system that we look at? And it comes down to the fact, I think, that we have dangerous prey. So there's an ability of some prey to generate a more severe consequence than just losing dinner. And that's what brings us to newts and snakes. Really, we're not going to talk about newts and snakes so much as we're going to talk about this molecule, tetrodotoxin. So at the core of this phenotypic interface is a toxin in the amphibians that we study. I'm not a chemist, but there's a part here that binds into the sodium channels in nerves and muscles, and then a part that acts like a cork and prevents sodium ions from moving back and forth across the membrane, and that stops action potentials. It's really well studied. We got lucky here because the thing that we're studying is well known from a neurophysiological perspective. It's got really broad toxicity. Every vertebrate has essentially the same structure of sodium channels. Most invertebrates have the same structure. It's found in many different taxa. So originally identified in pufferfish. It's known in blue ring octopus. You probably know this already. We don't know where it comes from. This is probably my biggest bugaboo when I try and figure out what's really going on is I sit around thinking, well, where does it come from? We don't know that it's genetically based. I told you we have to know it's genetically based or we're not really talking about coevolution. In the marine animals, it's thought that it comes from symbiotic bacteria. I won't have time to go into the data, but everything we know suggests that it's not a symbiont, it's not a dietary component, that the newts probably make TTX themselves, but we can't nail that down yet. So here are our players. Most places I have to go through a long, involved explanation of what this is and why it's super important. I can probably skip this. So here's Tarica. This is granulosa. You've probably seen them. You've seen Turosa. They're hard to tell apart here. If a bird attacks it or if a herpetologist messes with it, they go through this Unken display, and they're advertising toxicity. They're advertising tetrodotoxin in this case. So any animal that does something like this, you should know, don't mess with it. What got us onto this originally, or what got my dad onto it back when he was a undergraduate student, really. He started this as an independent study, what we'd call independent study now as an undergrad, was the observation that they were so ridiculously toxic in some places as to kill as many as 20 people by mass. We can quibble with the numbers, but they are definitely lethal to humans. So this works. This is my collaborator.
Probably the most poisonous animal in the world, with enough skin toxin to kill tens of thousands of mice or perhaps 100 people. Of course, they don't bite, so this isn't really dangerous holding it unless I were to eat it, which I won't.
So that's the message of the talk. He still doesn't know that I do that occasionally. But that's the point. You have to ingest the toxin. It has to get into the bloodstream. You guys know people handle these all the time. Half the people don't even know that they're toxic. So the other side of the coin is Thamnophis ortalis. This is a pan-North American species, at least as it's defined right now. These are 4 different subspecies, I think more appropriately color morphs from the western U.S. What's germane at this point to know is that they all are known to eat Tarica, but this is not a specialized interaction. We're not dealing with a situation where snakes from the Willamette Valley of Oregon eat mostly newts. We have literally 1 or 2 diet samples that have newts in the diet in each of these populations. We know that they are resistant. We know, we'll talk a lot about that. We know that it's a patchwork, it's variable, so it's not fair to say that all garter snakes are resistant or non-resistant. We'll talk about geographic variation. And then what's come about in the last couple of years is the realization that we were naive early on and that there are many other garter snake species that seem to have some resistance. And that becomes important as we try and tease apart issues of convergence and repeated evolution. Right? So some of you have seen this before. This is what snake biologists do. They belly flop on things and act a little more macho in the collection than your average botanist does, but that's what attracts people to the business. And then because snakes eat their prey whole, it's easy enough to find out what they've been eating. You perform this procedure known as barfing the snake, and you can figure out, at least within about 24 hours, what they've eaten recently. So this is an animal from the Corvallis area of Oregon. We didn't set this up. Everybody thinks they did. We were ready to set it up. I won't kid you, but that was a real find. So we do this in various places. We try and figure out diet differences to tease apart this ecological side. So that's the sort of setup of the natural history. And our problem when we got into this was, how do you study variation in something that is kind of a yes-no variable? They're resistant or they're not, they're toxic or they're not. And traditionally the way people study toxicity or resistance is through something called an LD50, the lethal dose that kills 50% of a sample. So you grab a bunch of mice, you shoot them up with a dose, it doesn't kill any of them, you up the dose with another group of mice, and you iteratively do this until you find a dose that kills 50% of your sample. And what you're left with is a population measure of toxicity, or if you did this to snakes, a population measure of resistance. And that doesn't work very well for teasing apart evolution, which is really an issue of variation. So we developed a bioassay based on sprint speed. So this is my laboratory assistant. He's racing a snake down a racetrack. We usually use neonates, although nowadays we've taken this up to adults. A snake will crawl down this racetrack, trip infrared beams, goes through a computer, you inject it with a known dose of tetrodotoxin, you wait 30 minutes and you race it again. When snakes can't crawl, they typically do more defensive anti-predator postures. This animal's going to now, very, very frightening, is going to bite the finger. When they do crawl, they're much slower, and we can get a quantitative and pretty fine-scale, very repeatable assay of how much locomotor deficit is generated by a particular amount of tetrodotoxin. So this one's barely able to move. So the bioassay is based on speed. We can use known doses. There are a couple advantages of this. So everything that you see from now on that's called resistance is going to be a measure of essentially percent baseline speed. 100% is the same speed that they crawl before an injection. 0% is flaccid, immovable, can't do anything. We like the assay for a number of reasons. To me, it still goes back to the ecology. If you pick up a snake that's eaten a newt, like the one that's belly flopped on in the video, there was no need to do that. If I'd known it had a newt in its belly, I could have walked up and poked it with my foot. They literally can't move for a couple hours after they've eaten these newts. So this is really the first effect of the toxin that we're looking at. It's the locomotor effects. They're repeatable, we can do it over and over again, they're very consistent, they're individually based, which means we can look at the genetics, and we can look at variation in populations that haven't evolved resistance. So we can go to a place that snakes have not co-evolved with newts and ask questions about variation that we could not do if we had to use an entire newt as the source of toxin. So what have we figured out? Over the years, these are some of the high points that I think set up the stage for what we're talking about in a minute. We know that there's a genetic basis. I'll dwell on this more towards the end. There are trade-offs with resistance, at least we think so. We've got a clue as to the mechanism, and this really broke open for us an understanding of some of the things that drive the dynamics. And then to me, still an underappreciated, but maybe the key to the whole thing working is understanding the behavioral interactions that allow selection to happen. So these are just some high points of older work. What's the genetic basis? We'll talk about the specific gene in a minute, but we started looking at heritabilities, essentially the percentage of genetic variation that's additive genetic in origin. These are really high numbers, 70 to almost 90%, but they're consistent with everything else we know. We know that we can't affect resistance through repeated exposure. The most extreme thing that we've done here is inject snakes every 2 weeks for a year, no change in resistance, so there doesn't appear to be any sort of plasticity or development of resistance. We'll talk about those genes in a minute, but the kind of classic data that we've got, don't ever try to breed snakes for a dissertation. It's a bad thing to do. So this is the result of several hundred adults and about 60 crosses, and we get 4 hybrid families. Right? But the point is, this is a resistant versus a non-resistant population. The purple bars are the hybrids. They're intermediate in resistance to the 2 parental types. We did the same thing with 2 resistant populations, one from California, one from Oregon. We get hybrids that are more resistant than either parental type. Strongly argues for a multiple gene or non-additive multi-allelic basis to resistance. So I present those mostly because they're the sort of intuitive genetic basis plot for most people. We know that there are trade-offs. These are representative of a bunch of different populations. These are litter mean plots, so every dot here is a family of snakes. Resistant populations and non-resistant populations show a negative trade-off between speed and resistance. And if you're still awake 20 minutes in, you should recognize that these axes should be autocorrelated because this is based on speed. So the correlation's a lousy way to test it. We've used other models to verify it. But again, this is the intuitive way to present it. There's a really strong relationship between the resistance level of a family or an individual snake and its potential speed. So we see it again, at individual and family level. We see it in resistant and non-resistant populations. that is important to me because it tells you that there's something more fundamental about the physiology of snakes, and that it's not something in particular about the resistance evolution that's driven that cost. It's something more inherent. And we think it may have to do with the mechanism, but this is getting past my knowledge of neurophysiology. We'll come back and do this. So what is the mechanism? So a few years ago, Shannon Gaffney, who's now a postdoc at Stanford, said, oh, I can test this hypothesis that sodium channels are resistant. And she goes and does essentially a textbook neurophys prep where she pulls muscle out of the snake, pins it down in a saline bath, puts current through it, and looks at the action potential rise rate. And her advisor said, oh, that's not really interesting. That's the prep that everyone's done already. But from an evolutionary biology perspective, we're getting a mechanism and we're able to find individual variation that drives what we see at the population level. So here's a resistant population from Oregon in saline and in a high dose of tetrodotoxin. The tetrodotoxin slows down the rise rate of current across the membrane. This is a resistant population from California. This is up near the Russian River, hit with the same dose. No effect at all on the action potential rise rate. If I showed you a mouse prep or a human muscle prep for this, it would be a flat line in the dotted line. There's absolute complete arrest of sodium channels. So even though there's a decrease in the action potential rise rate in these resistant populations, it's still way off the charts compared to most vertebrate channels. So this broke open again for us that it is indeed the sodium channels. She was also able to figure out that there is individual variation in populations that at least gives you the potential for selection to act on that mechanism. So instead of having a fixed physiology at the species level, we're looking at individual variation that might be the material that selection works on. And then still what I think is the most important piece from the ecological perspective is work that Becky Williams did as a master's student in my dad's lab. So she did what everyone thinks you should do. What happens when you put a newt with a snake? Right? So just to step back a minute, the paradox, when you have a deadly toxin or resistance to a deadly toxin, is it's very hard to figure out how selection acts at a quantitative level. So if you're a toxic prey, it doesn't do you any good to kill your predator if you die in the process. There's no variation in fitness. If you're a resistant or a non-resistant predator eating something that's deadly and you die with every individual you ingest, there's no variation in fitness, there's no selection. So what drives the evolution of toxicity to the point that it can kill a single predator, let alone 20 or 30 predators? There has to be some behavioral mechanism that allows for a quantitative relationship between toxicity and mortality. So Becky's experiment was to take newts from a more toxic population than the snake population should be able to handle, see what happens. You can do some statistics and it tells you that resistance determines how long they hang on to a snake. But the most important aspects from the study though are that snakes grab the newts, hang on, and then as much as an hour later reject the newt. So they open up their mouths and kind of back off, and the newt every time walks away. So head in the esophagus of a snake for 50 minutes doesn't seem to bother a newt. And this, if you go back through the old natural history papers of all kinds of distasteful prey, they all tend to be really thick-skinned and really durable and tough enough to be chewed on by predators. And that probably has something to do with the evolution of these distasteful secretions and toxic secretions. On the other side of the coin, the snakes that did that were immobilized, so they're still getting the effect. They're just not getting the full-on effect. One animal never did recover, so it rejected the newt. It didn't ingest it all the way, but it still had enough toxin absorbed through the esophagus that it effectively died. It was euthanized after 24 hours when it never recovered. And the one thing it predicts whether or not a newt is eaten is the resistance level of the snake. Now, we didn't have toxicity data at that time, so we expect that that's also part of the puzzle. But this establishes this behavioral context in which selection can generate this quantitative relationship between toxicity resistance and fitness of both species. So I still think this is one of the most important pieces of this whole puzzle. Anyway, that's my bit. So what do we do with all that? So there are essentially two major directions that we've tried to push. The first has to do with something that John Thompson's pushed now for, gosh, it's been a while, 15 years now? This idea that he calls the geographic mosaic of coevolution. I think it's more fairly a perspective, and it is just playing off the realization that populations are subdivided, individuals interact, that individual interaction is the ecological force that drives coevolution. So we see something happen at a population or species level because of what happens among individuals. Those interactions and their consequences depend dramatically on the details of the ecology of every single place. So if it happens in the Russian River area just north of here, or it happens in British Columbia, the potential predators there, the parasites, alternative prey base, growing season, all those things differ and are going to drive different outcomes to coevolution in each of those spots. Thompson predicts as a result of that we get coevolutionary hotspots and cold spots, places where reciprocal selection is intense and where reciprocal selection is non-existent. That's prediction one and the primary piece of that story. When that holds, there are a number of hierarchical predictions, general predictions that come after that. One is that we should see places where there's polymorphism or a lot of variation maintained because we see a selection mosaic and individuals move back and forth moving genes. We should then find places where there are unmatched populations, places where, say, a resistance gene is being moved through gene flow and you get areas where the predators are better suited, more extreme traits than need be, something that some people call maladaptation. I'm not a fan of the word myself, but the notion that functionally there should be places where they are mismatched. And if all that happens, then you can also make this general prediction that the evolutionary trajectories or outcomes are going to differ from lineage to lineage. If we go to one group of populations, we may find one set of outcomes in terms of the status of the arms race. We may find another outcome in another place. We may also find that the genetic mechanisms or physiological mechanisms differ from place to place. So we did this, again, to remind you what data we're working with. We're racing snakes, injecting, and looking at percent baseline speed. This is the kind of information that we get. So here are five representative populations. Purple and blue are what we would call non-resistant. Ecologically, they're not resistant enough to handle a toxic newt. Yellow is kind of a low-level resistance. Orange is pretty high resistance. And then we've got these crazy things out here that are 3 orders of magnitude more resistant than the ancestral state. So we can get a dose-response curve. We can interpolate a 50% level. So we show a 50% dose that knocks the average snake down to half of its crawl speed. We do this for a bunch of populations throughout the western US, and you see that there's just this tremendous amount of variation. This is a log scale down here. The units are called mammoos. This is another cost of collaborating with your father. He invented a unit years ago that I don't like, but it's milligrams of tetrodotoxin per gram of snake, essentially. So and then translated into how much it takes to kill a mouse. So think about it as a mass-related dose. But 1 mouse unit per gram of snake knocks a snake down to 50% of its crawl speed in these populations, 1,000 units up here for these populations in red. So this doesn't tell you much about geography. You put it on a map. Here's the range of Pterica, the genus, in gray. We've got populations in allopatry, populations in sympatry. When we started this, I thought we were going to go out and prove the obvious that we had non-resistant populations here and we had resistant populations in sympatry. I'd be done with this and I could go on to something more interesting. I was fortunately very wrong. So there's a lot of information in a plot like this. And the first thing that you see is this low level of resistance. But the second thing is that you see 2 apparent hotspots, one up here in Oregon, and one down here in the Bay Area. And in between we get cold spots or low levels of resistance. pretty much low levels of resistance in the Sierra, and as you go north, low levels of resistance. And if you plot that out and make an isocline map, it makes it a little more visually appealing. Again, you see these centers of resistance evolution and clinal radiations out from there, which you can spin two different ways. You can argue that there's variable selection along those color gradients, strong, weak, weaker, non-existent, or you can imagine that there's only selection in those hot spots and export of genes out. So they're consistent with several aspects of the geographic mosaic, but not demonstrative of either one. All right, so why do we get this mosaic? And it's admittedly, I think what I ask you to focus on are just these two main areas and realize that there's more variation out there that's of interest. So the first question was simply, why do we see two places? Did we get one origin of a trait and different invasions of Oregon and the Bay Area from a common lineage, or are these truly independent evolutionary origins? So we teamed up with Fred Jansen's lab at Iowa State, took a subset of these populations. He generated a mitochondrial phylogeography here. For those of you focused with the California view of the world, this is not a very satisfying tree, but it's really what happens when you go to the Pacific Northwest. This is the conservative version of this, so these are bootstraps of, I think, 80 and above. We get three clades. We get one that's Pacific Northwest, we get one that's sort of Intermountain West here, and out to Idaho, and then we get a nice, well-behaved California clade. There's not a lot of resolution. There are not a lot of informative sites in this tree. What we wanted to really find out was how did the resistance traits map onto the tree. We get two clear origins of resistance, and probably a third over in the Sierra, but that's putting a lot of weight on a branch that I've collapsed here that I think doesn't have tremendous support. So we have at least two, maybe a third origin of resistance. And that's partly, I can make that statement partly because the outgrouping in the ancestral state is so obvious in this group. So it argues that, in fact, we've seen coevolution or some evolutionary force drive resistance at least a couple of times. So you want to know why. We assume that it has something to do with newt toxicity. This is the only plot I'll show you of actual data from newt toxicity. This is Charles Hannafen's work. He's a postdoc at Hopkins Marine Lab now working on channels in newts, salamandrids writ large. This is an HPLC plot. He pioneered this whole technique, or brought it from Japan, made it work. It involves a lot of spilling of chicken blood and alcohol sacrifices. And then you don't ask why it works, but it does some days, and then other days the machine doesn't work. You get a peak. Area under the peak gives you the amount of tetrodotoxin. There's nothing here, and there's nothing ready to pop up. This is Vancouver Island. There is no detectable tetrodotoxin in that population. And that's true for many populations. If you look at the resistance levels of the snakes, they match up really nicely. You should stop when you get 2 points. Nice correlation, spectacular. We're not that smart. So Charles, over the years, collected toxicity data on many, many populations of snakes, about 30. And this is what we came up with. This just came out, I think, last year. And there's a lot of explanation that goes on here. So these are now levels of toxin translated up to a whole newt, whole Tarica, whole Thamnophis sertalis. They're based on size, and that's why you see negative numbers. This solid line is the 50% dose. So how much does it take to knock down an average adult-sized snake? That's right here in the solid line. That's the 50% line. And then above and below that are two lines that correspond to the 15% dose and the 85% dose. So below this line, you get a region where no snake is affected by a newt with this toxicity. Essentially, a snake could eat any newt in this range and have no detrimental effect on its performance. Above that line, you've got the converse. You've got any snake eating a newt with that much toxicity is going to end up dead. So these cross plots, and there are error bars here, they're just tight in some places, You can do this with the range, which is really the proper way to do it. I just didn't have that plot. They fall fully outside of that range. And that means no newt in that population can affect any of the snakes that we measured in that population. Essentially, there's no potential for reciprocal selection. In this band, we have the potential for reciprocal selection. There could be a lot of things that go on that make that not happen, but just on a functional ecological match basis, we think that that zone is a place where reciprocal selection could occur. Those populations in orange correspond mostly to the northern part of the range. I don't know why. The populations that are what we call mismatches all fall on this side of the line. In no cases do we get prey too toxic for predators to handle. In every case, we get predators that have escaped the arms race, that have become more resistant than they need to be to handle the local prey. And there are two kinds of populations there. Down here we've got populations that seem like they never really got started. The ancestral state for Natricine snakes, for garter snakes and their relatives, is right about here. So it's a little bit more resistant than they need to be to handle a very low toxicity newt. The ancestral state for newt toxicity, if you base this on other genera from Europe and Asia, is right around here. Right? So these populations have no elevation of resistance. And they correspond mostly to the very northern populations, but also some of the southern populations of Turosa, some of the Granulosa on the north coast. Then we have this third group. These tend to be in the Bay Area, with one exception over here in the Sierra. These are all cases where we have very toxic newts, toxic enough to kill snakes from other places, and really resistant snakes. Snakes that are so resistant that we've never found a newt anywhere that could have any effect on those snakes. So it seems like they have literally shot out of and escaped that arms race. I'm not willing yet to say that that's happened twice, but I think when we get better genetic information, we're going to find that this Sierra lineage is in fact a different origin of resistance and a different allele. So apparently once, maybe a second time. Says the same thing. The point is that there are two kinds of snakes being ahead of newts. So if we go back to the geography, this is where we were a few years ago. This is where I was a few slides ago and said we have two hot spots. We know that resistance is highly elevated. This is kind of the, I guess the message of this slide is the pitfall of assuming too much based on phenotypic patterns. So if we look at resistance in snakes and we compare it to the geography of newt toxicity, you see elevated traits in both species in Oregon, you see elevated traits in both species in the Bay Area and maybe in the Sierra. If you just compared those two, you'd say, we've got strong coevolution here, weak in between, and weak to the north. But when you evaluate it from an ecological perspective and ask functionally, well, that's a lot of toxin, but is it enough to knock down a snake? You find that the answer is yes in Oregon. You do the same for the Bay Area, and you look and you find the answer is no. So the pattern of mismatches is very different than the pattern of elevated traits or even the similarity in the pattern of traits. So the message here is that to evaluate reciprocal selection, you can't rely on patterns of phenotype. You have to go back to some sort of functional interpretation. And this is admittedly a pretty crude napkin math kind of way to do it, but it's the best that we've been able to come up with so far. All right, so in general, what does this show us? I think this is the first time I've really been satisfied that we have evidence of something akin to an arms race. And now this is storytelling, but this is what it seems like is going on to me. We start populations down here with the ancestral conditions, and that's just a statement based on the comparative analyses that resistance is low, but a little bit higher than it needs to be. Snakes are ahead. Newts really shouldn't be responding to selection, so something else drives them to higher levels of toxicity. Maybe it's another predator. I think probably is another predator. When they gain this zone of reciprocal selection here, you start this arms race interaction and they essentially climb that climb, and they can wobble around. I don't know what ability there would ever be to predict whether you're above or below a 50% dose. That's kind of an arbitrary position for us. And then at some point in their evolutionary history, something, in this system at least, causes snakes to escape. They get out. And they get out because of some, I hate to use the word, but saltational evolutionary step. They have moved so quickly from this level of resistance to this level of resistance that they were able to lose the tracking of the prey. Now I show this to different people and they say, well, that shows an arms race, but why wouldn't costs then drag those localities back either down here or back into the zone of reciprocal selection? And I think that's a reasonable hypothesis, but we don't see any populations in that zone that I think you would be likely to pick up if that was what was going on. So is it cyclical or is it an arms race? Our data seem to suggest arms race. All right. So what do we do with this information, and what can we tell based on a more reductionist approach? So we see that all these mismatches go in one direction. Is there something about the mechanism, the genetics of it, that helps us explain that? And I'll argue yes. Can we pick up patterns of convergence, and can we use that information to ask things like, why? Are these new novel mutations? Are we weeding out standing genetic variation? Have genes moved from one population to another through integration? We're starting to get the genetic tools that we can ask those questions on. And now we have multiple species that we can ask the same sort of thing at another level. Okay. So now we're back to Shanna and her work. She cloned out the sodium channel gene in skeletal muscle. It's called NAV14. This is a cartoon version of it. Here's a membrane. There are these columnar pieces that create a pore. There's a part that's outside the membrane, a part that's inside the membrane. These are duplicated regions, and this whole family of genes has been duplicated in vertebrates. So there are 10 in humans. We don't know how many in reptiles, probably 6 to 8. Everything we know from site-directed mutagenesis studies suggests that this is the region that should generate binding to tetrodotoxin, domain 1. We see no action in domain 1. What we found was changes, substitutions in domain 4 that we think are important. So here's a non-resistant snake. Here's an amino acid sequence from domain 4, this little piece that sticks out and is bound by the toxin. This is the same amino acid sequence that you have, or a fish has, or a bird, or a mouse has. It's incredibly conserved across vertebrates. If you go to a mildly resistant Oregon population, you get one substitution of a valine here. You go to a slightly more resistant population, you get the same valine substitution and another one over here in pore. And then you come back in the tree, it's not clear from this picture, but you come back to these California groups that evolved independently, you see 4 amino acid substitutions. But you see the same valine, and that probably arose independently, at least if our phylogeographic hypothesis is accurate. Corresponds nicely with our levels of resistance. Seems like we have something that functionally is related to resistance, but Shanna is much better neurophysiologist than we'll ever be. So she goes and does what I consider still voodoo. She is able to take these genes and first she's able to take the genes and do site-directed mutagenesis and generate a human snake chimera. So she takes a functional human sodium channel gene and changes just these amino acids and says we can now test the functional relationship of resistance and those amino acid substitutions outside of the context of the snake gene. And we're going to take that gene and we're going to put it in the xenopisthocyte so it's in a different genetic background. She did that for a chimera and then took the entire snake sequence that we had to ask whether there are other aspects of the gene we're not focused on. And here are our 4 populations again. One of them's green. It's kind of, this is Warrenton that doesn't have a curve. What you need to see from this is that on a crude level, the resistance level of that channel, that gene expressed in the xenopisthocyte, matches very nicely our population differences and our whole muscle differences. So the genetic level seems to pair up nicely with our whole animal biology. The chimeras that she made had the same behavior as the full snake channels. A little bit of a difference, in these two populations, but not much. And that suggests that it's in fact these substitutions that we're looking at, not something else. And then one of these serendipitous screw-ups, she forgot to change one of the amino acid substitutions in one of her chimeras. So she left out the one that's most common, this valine. And she found that the valine has very little effect. And I would have looked at this and said, it's the one that's most common, it's arisen twice, that's likely the magic bullet. She found that it's less than a twofold difference in resistance, where we have a 3 order of magnitude, 1,000-fold difference in resistance to explain phenotypically. So using this approach, I think we can get further at which one of these amino acids has the biggest effect and whether we have non-additive substitutions, whether we need the valine for the others to have an effect, and so on. Look at order of accumulation of mutation. So that's our tool. We've got a genetic mechanism. We see some level of independent evolution in these lineages. They're doing it somewhat differently. They're accumulating different amino acid substitutions. Now we try and go back to the species level. So here's a tree of natricine snakes, mostly Thamnophis. Sertalis is what we focused on. For the longest time, we thought they were the only resistant species. And then people started sending us reports of a couchi ate a newt here and an Atreides ate a newt here. And it turns out that there are at least 3 other western Thamnophis that in some places are resistant to toxin. Couchi, in most of the places in the Sierra, Atreides, at least in the southern Bay Area, Monterey, Gilroy area. This is a messy plot. Here's the ancestral value for the subfamily natricines. Here's a plot for Thamnophis sertalis from Benton County, just as a marker, kind of. And then these color-coded things, these are Atreides. We've got non-resistant populations, resistant, super resistant Atreides. We've got low resistance and super high resistance couchi. We've got pretty high resistance elegans. The point is simply that we get this whole range of variation in every one of these species. And the reason that we thought none of them were resistant was we had only sampled these guys. We, kind of embarrassingly, we've been focused on Oregon. We didn't look at California until too late. And that's when we figured out that that's where all of this action was going on. So here's where I really throw up my hands. Species are different symbols. Colors are different levels of resistance. What's the geographic pattern? How can you make any sense out of this? The best I can do right now is draw big circles and say none of this taxa are resistant at a very high level in Northern California. None of the newts that we've sampled in Northern California have very high levels of toxicity, and newts are not found in some of these areas where we've sampled to the far east. Then in the Southern Sierra, everything is resistant. That everything being couchi and sertalis in one locality of elegans, but everything seems to be pretty highly elevated. The Sierra, Tarica Sierra here, they're not super highly toxic, but pretty toxic. And then, I don't know what's going on here. You can literally go 10 miles and find 100-fold difference in resistance between Atreides populations, going from Monterey up the peninsula, or just going over the coast range here from Carmel Valley. Carmel Valley's kind of in here. Anyway, you get dramatic differences just over a little bitty geographic range. We still don't have a very good handle on this. We do know from work that Chris Feldman's done that we have several different alleles for resistance up in here. And maybe that what we're seeing is movement of genes from population to population. But we see 2 places where we have very general concordance in geographic pattern, and 1 place where we see nothing like that. So the last point is, can we tell anything about convergence? And this is Chris Feldman's work. He's sequenced a phenomenal number of snakes for the entire sodium channel gene. Here again is the cartoon, domain 1, domain 4, domain 3. Here's Sertalis from Willow Creek, a very resistant snake. And on the right here, we've got the mitochondrial DNA phylogeny from DeCaro's work. This is species relationships. And over here, this is Chris's NAV14 tree based on non-coding sequence in that gene. You can see that it's a pretty close match. And we've got differences in amino acid sequences. So in a Tradeoffs, we've got one common substitution in domain 4, and we've got 2 novel substitutions in domain 3. These are not found in anything else we've looked at. They're not found in Sertalis. He can argue based on site-directed mutagenesis work. So the neurophysiologists take the gene, they bomb it with mutagens, then they see how it binds to tetrodotoxin in a mouse model system, and they say that this is a position that seems to be involved in resistance. And that's in fact what we're basing our functional relationship here on. You go down to Cauchy, right? Where is it? We've got one change in domain 3. It's not the same as the change in a Tradeoffs. It's not found in anything else. It's not found in Sertalis. So we're getting different changes at different domains. We have no changes in domain 4 for Cauchy, and yet we've got populations that have an 80 MAMU resistance level, as much as any of these other things. I think the most impressive thing that Chris pulled out of this, to me, is the argument that this completely puts to rest the idea that there was standing genetic variation in sodium channel 1,4 that was sorted out. So if that was the case, you'd expect these alleles to come out in a sodium channel tree close together, and they don't. So pretty clear evidence that we do not have standing genetic variation being sorted, that these are novel mutations, new generation of variation that selection's worked on, and that introgression really doesn't have a role in this. So moving genes through rare hybridization across species, we'd at least expect to see the same alleles. So, wrapping up. Geographic structure is critical. I still can't tell you exactly why or what all the nuances are, but it really is obvious that if we aren't looking at a population level, we're going to miss a lot about the dynamics. The little bit of crow that we had to eat based on an earlier publication is that our phenotypic hotspots don't reflect coevolution. And I think this is a good lesson. It's been emphasized by Richard Gomulkiewicz and Scott Neusmer and others in a recent review that says you can't just look at phenotypic patterns and make arguments about coevolution and geographic mosaics. You really have to get deeper into the process. There are places where traits are so mismatched that we think current coevolution doesn't happen, that reciprocal selection is obfuscated by the degree of functional mismatch that we see. It may be that there are other aspects of coevolution going on, or that if you take the long-term view and think about costs, that coevolution is not done. But right now, there's not reciprocal selection in some of these places where the snakes seem to have escaped. To me, the most fun's been getting into this genetic basis of resistance and asking about the functional changes. The problem is we don't know anything about toxicity. So you'll notice in the beginning I said something about reciprocity. We don't know coevolution unless we study 2 species. All the data are on snakes. We still don't know much about newts. We don't know if newts make their own toxin. So someday I hope we can get down to this level and know something about that gene for gene evolutionary interaction. And then finally, because we've got multiple species that are independent, I mean they're distant in that garter snake phylogeny, we can ask about convergence and parallelism. And I argue back and forth with Chris and Mike Frender, the people that we work with on this project, what's parallel and what's convergent. You know, it's the same gene, but it's a different part of the gene. It's different changes. Does that make it parallel or convergence? It doesn't really matter, I think, except to know that you've got the same gene being manipulated by evolution through novel mutations. So those are the main messages I leave you with. And don't eat newts. That's key. So lots of people work on it, grad students in my lab. John Chekalochik is the guy who's running the molecular operation in my lab right now. I don't know a bloody thing about it. Chris Feldman just finished at USU and has a spate of beautiful papers like that last slide. Jeff Mitochak did some of the original phylogenetic comparisons. Becky did still my favorite part of the ecology. And then other collaborators and people gave us money. And thanks for listening and inviting me back.
So I think some people may have to run off to
away.
classes, so we'll wait for a moment, and then those of you who'd like to stay behind and ask questions. I'm gonna let Butch field his own question.
One of the things that seems to me to be important is to know how often these snakes eat a meat. For example, in those areas where there's big mismatches, maybe they're smart enough never to try to.
Yeah. This has been the frustration to me from the get-go. So, and it has more to do with logistics than anything else. It's hard to get really good diet data. So you can go out there and collect 100 snakes in a week and you get 20 or 30 diet samples. And my field work and the field work of anybody in the project is really constrained to short windows of time where I can go out in May or I can go out in August, but the newts are most active at a different time of the year. Maybe there's a different life history stage that's important. It's absolutely the critical data, and we lack it.
And to sort of extend that, so there are places where you have more than one species of Thamnophis, and so I'm always a fan of historical explanations, and it may be that if one species of Thamnophis was there first, it evolved some resistance, whereas the second species may be a more recent arrival and just hasn't had the time to evolve the sort of resistance that it would need, or maybe it doesn't need the...
And that was exactly what we were hoping to get at here. We don't, so far at least, it doesn't look like we're going to have the resolution to be able to say Couchi was here first. And that's what we had kind of hoped, was maybe we can do some sort of historical precedence. The first species in evolves resistance and the others are left behind. And maybe that's some part of what's going on here. I think it may have more to do with syntopic ecology. Are there places where a Tradeoffs really lives right where the newts are breeding and the new newts are coming out, new metamorphs are coming out, or is it Sertalis?
That was my question actually, because you have elegants eating different things in different places. They're definitely eating aquatic stuff in the Sierras and more terrestrial prey in the Bay Area, and then the newt activity differs wildly, on the north and the south.
I mean, you come out here, you find them in rivers, and you find them in little creeks up in the Sierra. The biology is enough different that I think that stuff matters a lot.
So have you brought any of that?
Not in any systematic way. Still, the thing that drives me crazy is we don't have that seasonal information because nobody has been out here to devote a year-round field work effort.
But I think related too, what do you know about the ontogeny of both the resistance and toxin, and is there experience? Can snakes become resistant?
They can't become resistant. There's an ontogenetic effect that's a size dilution effect. So a big snake is more resistant than a small snake. But it's really just a size thing as far as we can tell. The toxicity development in the newts is kind of, I think there's more to it there because larvae are not toxic. Eggs have toxin, larvae lack toxin. Sometime between metamorphosis and adulthood, they become toxic, and we still don't know what point that is. But certainly a little metamorph this big has much less absolute level of toxin than an adult. And we think in places like Molino and the peninsula here, you've got relatively small Tradeoffs that seem to be eating small metamorphs. And I think that you get started there with low levels of toxicity.
I had one comment and then a question. My comment was that when you first showed your map, it was really striking to me that you have these two major river systems where there seems to be hot spots. And even Monterey, that the river system used to come out there. And then in between you don't. So that was just an observation. But I was wondering, related to that perhaps, is there some other alternative prey in those big river systems in the valleys and the peaks up that would also have Chicototoxin?
As far as we know, nothing else in this part of the world has it. It's found in a variety of other amphibians, but nothing else here. There's a related toxin called saxitoxin that's produced by some algae, and resistance to saxitoxin and tetrodotoxin and other things seems to be parallel. But
so would there be something that other prey items would be picking up from the environment?
Nothing that
From
we've
those environments?
been able to put our hands on. It's a very weird molecule. It's not very similar to any, it's a guanidinium, which doesn't mean a whole lot to me, but it's a very unique structure that isn't, it's not an alkaloid that is similar to other things that they might encounter. So it's a pretty unique thing. Now the other thing that I glossed over, it's kind of the other part of the side of that coin, is newts have a lot of other stuff in the skin. We treat TTX like it's this magic bullet, and there's a lot of other stuff that can be just as important.
Has anyone tried raising newts inside their unit?
Yes. Stupid project 32. Yeah, it turns out to get an egg to adulthood is about 6 years. So out of the 300 eggs I started with, I got 6 adults. They seem to have tetrodotoxin, but not very much. So it's again this kind of unequivocal answer. It's they have it, but they don't have as much as you think they should have.
One of the things that struck me from the graph when you plotted the resistance versus the, I guess, the lethality, there wasn't much variation in the newt compared to the snake. If you looked in other species that had TTX, do you see that little variation across?
There's actually quite a bit of variation, and it's probably the graph that I had up there is not got the best bars. So, let's see if I can do this to hop. This one?
Yeah.
So these are standard errors, I think, or 95% confidence intervals in this plot. If you do the range, it's a little bit broader, but there seems to be less variation in most of the resistance data than there is in the newt data.
Oh, I meant across population.
Oh, across
And
population.
there's much more on the
Yeah,
x-axis
yeah.
going than the y-axis if you look.
No, there certainly is, because you're talking about an order of magnitude scale here and a more linear scale here.
Will give you any insights of how you get it.
Well, the one kind of, again, napkin math thing we did was to move these snakes up into here, you'd have to get 10% by dry weight toxicity in the skin for a newt. So they would have to have